Hey everyone. So I'm not a student. I work at Arcus Biosciences, but I'm definitely a student of HDF5. Probably I would know least here because I just heard of the technology a couple of months ago. Um, so essentially, we want to leverage HDF5 for immuno-oncology, and I think one of you I believe, also spoke of bioinformatics as one of the workflows. So this is exactly that use case. So immuno-oncology essentially means you're trying to trigger an immune response and cure the cancer. And we also work on designing combination therapy. So there's like partial chemo and our drugs that can help with the immune response for cancers. And we are working on multiple different cancers, like colorectal cancer, lung cancer, etc. But yeah, so let's bring to the. So our use case, why we need HTTP5 is we're looking for something that's like a fair data storage, interoperable, and accessible, usable principles. And one of the best part about this is if all the metadata, if everything is self-describing, this is one file or a couple of files, it's really easy to find with bioinformatics at the company to kind of work with their data. Right now, most of their data sets are like in S3, CSV, TSV, and then they join different tables and use a bunch of different sources. We want to collect everything from the source. So before we get into what's the specific use case, this is kind of like the drug discovery process. You would have seen that. And this step is essentially for pre-discovery and the targets, and then also the next software maybe that once you do phase 3 trials or even phase 1 trials, you want to try and validate whether what you did, all the predictions you did actually map to what happened when the drug was administered in the patients. So specifically, this is on the gene expression. Gene expression profiling is indispensable, as we know in today's. We have tremendous ability to predict about what would happen if you target specific receptors. However, singular genes are no longer what happens within a cell. It's an entire gene set, which means a lot of pathways are involved, like about 50 odd genes would be involved in a specific scenario. So there's a process called as gene set enrichment analysis, where you determine what genes are associated with a specific biological pathway. So essentially, we want to store all of this information for our research. What's the membership? Which pathway are the genes part of? Co-expression, which means that if some external target or stimulus comes in contact with your receptor, what all gets expressed? Multiple pathways can fire off, multiple genes can get expressed. There are certain gene families which always live together. Then there is phenotypical expression of what exactly, when you draw blood, when you do the blood profiling, what kind of phenotypical, that is like the actual expression of the gene is provided. We want data for that. We want the location because we need to know exactly where these genes live. If you try to attack something which is closer to some other sensitive area in your genome, that's an issue too. We want to know that. There's something called transcription factors that bind your chromosomes or the genes at specific sequencing locations, and those impact how the protein gets translated by the cell. So all of these information is important. I'm not sure if you can see, it's a little small, but this is one of the famous gene sets, and there are many such databases like BioPart, KEGG, etc. This is one of the famous one called as Hallmark gene sets. It has like a combination of different sets. We have some internal gene sets as well as we reference these that are publicly available. But essentially, if you see there's a lot of information here. There's like relation with other gene sets and what bibliography is part of that gene set. So all of that we want to store. So right now, like I had mentioned, wait, is that going to the next slide? You have to zoom out. Yeah, okay. So essentially what I talked about, we have a bunch of CSV and TSV, some Postgres table. We we merge all of that into Python data frames, R data frames, and try to do kind of like in-memory analysis, or we run it as like a job. But we really believe HDF5 would help us here. So we're still in design mode, full disclosure, but hopefully as we learn more, we'll share with you. We'll learn from all of you on how to do it right, all the things and all the gotchas that you figured. But that's essentially the goal. And we think that the grouping and linking is going to significantly help us because each pathway can have multiple pathways that can have its own metadata. And this is how we plan to kind of store it, that it'll go down into the gene level, and that table you saw in the hallmark gene set, we'll use that with our internal gene sets. That's about it. We have some more future plans, so we plan to store. That's part of the poster. Yeah, I guess that's it.